Hi, this is Professor Wang from Philip and Imad's Biolab World with Professors Tavan and Sam here. And basically today we are testing the levels of CO2 that are produced from cellular respiration from germinated and ungerminated peas. Now in this setup we have germinated peas on this side, and we basically filled this test tube up with water, submerged this tube in the water, and we capped the germinated peas to block out the oxygen. Then we capped the top, and now we're gonna wait an hour to see how much CO2 can gather up in and out. And we basically did the same for ungerminated peas, except they're ungerminated, so we know that the rate, we're hypothesizing that the rate will be slower.
The phenol. Explain the phenol red.
Later we'll be using phenol red. We'll replace the water with phenol red solution, which tests the pH. And then we will bubble the carbon dioxide with the phenol red, through the phenol red, in order to test and see our results about the rate of the CO2.
In the third part of the experiment, we have boiled chickpeas that
Professor I have just prepared.
Which he has just prepared all by himself.
Obviously. With other people.
No. And we're gonna put them in the same setup with a tube like the with a jar like this and the tubing.
Because they have been boiled, the enzymes
like
in these chickpeas have been denatured. And so our hypothesis is that respiration shouldn't be very efficient or in fact be able to take place at all. Which we should be able to detect by a lack of carbon dioxide and a lack of color change in the phenol red. What we're doing right now is we're bubbling the carbon dioxide that has accumulated in the jars of the seeds through phenol red. As you can see in this one, through which we've already bubbled carbon dioxide, the germinated seeds produce a significant amount of carbon dioxide. It's bubbled through this, turned into carbonic acid, which changed the phenol red to be a light yellow, which means it's acidic. Here, we just bubbled through, and the phenol red barely changed at all, which implies that not a lot of carbon dioxide was produced, so the non-germinated seeds don't really have a very high metabolic rate. Now we're going to do the same thing in a little while. In a little while, we're going to bubble carbon dioxide through the carbon dioxide that was produced by germinated seeds that were boiled, and the name enzymes of them denatured, through phenol red, and see how much carbon dioxide was produced by denatured enzymes.
Yeah.
We have now completed the experiment in which we measured the metabolic activity of chickpeas under different conditions. Here we have the carbon dioxide from chickpeas, from germinated chickpeas. Here we have the carbon dioxide bubbled through phenol red from ungerminated chickpeas, and here the carbon dioxide is from germinated chickpeas that were boiled, so their enzymes denatured. As you can see, a significant amount of carbon dioxide was produced by the germinated chickpeas, as evidenced by the color change of the phenol red. Almost no carbon dioxide was produced by the ungerminated chickpeas, which isn't surprising because the chickpeas are hibernating. Also, almost no carbon dioxide was produced by the boiled chickpeas, because that denatured the enzymes and thus their metabolism could not be very active. Strangely enough, if you look at this closely, the boiled chickpeas are actually, even though they're boiled, have actually been more active than the ungerminated chickpeas. We're guessing this is because that even though we boiled them and all the enzymes denatured, after we cooled them down back to room temperature, some of the enzymes might have renatured, so we got a little bit of metabolic activity.
Welcome to Philip and Imad's Biolab World. Today we've got the assistant professors Sam and Pavan.
Assistant professors.
Doctor.
Herr Doctor Assistant Professor.
And today we're going to talk about our fermentation experiment.
Our fermentation experiment.
Assistant? Explain.
Well, what we did is Dr. Harris, assistant professor.
Here, doctor.
What we did is we took 15 milliliters each of 0.6% glucose solution, lactose solution, and fructose solution. We put about half a gram of starch into each one, and then dumped each one of these solutions into a swan tube. So what you do is you put liquids into here, close it, turn it over, liquid falls in here, turn it over, and let go. And because of there not being any air there, all the liquid is kept here because of vacuum pressure. Then we are incubating it for 37 at 37 degrees Celsius for half an hour and then an hour. And we're going to measure the size of the bubble of carbon dioxide that forms. This way, we can measure the rate of respiration of yeast, how fast yeast metabolizes different sugars, fructose, lactose, and glucose.
Now, Assistant Professor Pavan, do you mind explaining the fermentation process?
Of course not.
Notice how much these two do.
Our fermentation process in
Fermentation process in general.
general. Okay. Well, fermentation is a process that cells undergo during anaerobic conditions. So essentially what happens is, with the absence of oxygen, cells can only undergo glycolysis. So now the problem with glycolysis is there's going to be a buildup of NADH, and you need NAD plus to for glycolysis to occur. So how the cells get rid of glycolysis is through conversion of the pyruvate, the products of glycolysis, to lactic acid or ethanol in the case of yeast.
30 minutes have passed of our fermentation experiment. Number one, the swan tube number one is the glucose tube, and we've got a moderate amount here. Swan tube number two is the lactose tube, and as you can see, the lactose tube hasn't really had very much activity. Tube number three is the fructose tube, and as you can see, yeast metabolizes fructose very rapidly.
And we had these incubating at
So far they've been incubated for half an hour. We're going to incubate them for another half an hour and remeasure. We have incubated the yeast with sugars for another half an hour. So, as I remind you, this is the glucose, this is the lactose, this is the fructose. As you can see, fructose is very actively metabolized, lactose isn't very actively metabolized, and glucose is fairly actively metabolized.